All right, good afternoon, everyone. Thank you for coming to Nugen's workshop this afternoon. My name is Joe Don Heath. I'm the Vice President of Market Development at Nugen. We've got a series of talks for you today, a couple from customers, real live customers that have used our products, and a couple of talks from scientists from Nugen, myself being one of those. So I wanted to start off by saying that Nugen hasn't historically attended PAG much at all. In fact, I think this may be our first real official time that we've attended. And I have really enjoyed this meeting. I think it's been fascinating. There's just some great gains being made in knowledge in plant and animal genomes, and the technology improvements that are enabling these kinds of advances are really fascinating to see, and what they're enabling people to do. And what I'd like to tell you about briefly is how Nugen hopes and how we have developed systems that we hope will help you address some of those problems. So I should introduce Nugen because, like I said, we haven't been here. Nugen has been in the business of sample preparation for genomics for about 13, 14 years. We started off in microarrays, and then over the last several years, we've been building out a next-gen sequencing portfolio of library prep solutions. They fall into categories of targeted sequencing, and I'm going to be talking about our targeted sequencing solution today for targeted genotyping. But our targeted sequencing solution works with both DNA and RNA if you're doing targeted sequencing on your organisms. We have DNA sequencing solutions, DNA library prep solutions. The feature there is very low input with quantitation-free, titration-free, and dimer-free library prep. This has been a very powerful library prep system for people that are working with very small samples or ChIP-Seq, things like that. We also have an epigenetics library portfolio for methyl sequencing, both a whole genome methylation system as well as a reduced representation targeted methylation. And then finally, RNA sequencing. This is one of our largest portfolios. We offer a broad range of RNA sequencing library prep solutions for any species, any organism, and one of our speakers today is going to talk, two of our speakers are going to be talking about that. All right, well, I'll move right into what I'm going to be talking about today. Oh, I had one more slide here. And all of these solutions can be applied into a wide range of agrogenomics applications: SNP genotyping, epigenetic studies, whole genome sequencing, whole transcripts, transcriptomics, and also gene expression analysis. And you'll hear a few of those examples today. So let me start talking about our new targeted genotyping solution. This genotyping solution uses our patented single primer enrichment technology, which I'll explain on this slide how that works. We design custom probes based on your SNPs of interest, your targets of interest. We design probes near those SNPs so that every read essentially from your targeted panel gives you genotyping information. It's a very flexible design, and it's a very scalable design that can be scaled from hundreds to 100,000 SNPs or more in a single panel. And we can do this on any sequenced genome. We've worked very hard at improving the cost efficiency by creating a highly multiplexed library creation system and highly multiplexed sequencing. And I'll tell you a little bit about how all that works as well. So how does SPET work, single primer enrichment technology? So the first thing that happens is the DNA is fragmented, random fragmentation. This can be Kavaris shearing, or it can be random enzymatic shearing. We're developing our own enzymatic shearing method that is part of this system. So we fragment the DNA and ligate on indexed adapters to every fragment. Now you can see illustrated here with the red star, I've indicated a targeted SNP that we want to enrich out of this pool. Next to that targeted SNP is the region where we're going to design our targeting probes. So once we've ligated on our indexed adapters, we pool all of our samples together so we can multiplex a high number of samples at this step. We pool them, add our targeting probes, and anneal the targeting probes to their region, to the target region. The targeting probes bring in the forward adapter, whereas in the first step, what we've done is ligated on the reverse adapter. So the targeting probes now are bringing in the forward adapter. We extend those probes through the target region, completing the reverse adapter, and then those molecules, only those molecules that have both a forward and reverse adapter sequence, go on to PCR amplify. So this technology is unlike amplicon sequencing, because we're not relying on 2 primers per SNP. We're only relying on a single primer. That's all we need. It's also not a capture pull down method. We're not doing any sort of capture or pull down. So that's how the SBET technology works. A little bit about the workflow. This is a very simple workflow. It can be completed in less than 24 hours. Starting with 100 nanograms of DNA, there's a fragmentation step, followed by an indexed adapter ligation step, and then pooling and purification. This whole process is roughly about 2.5 hours. Takes about 45 minutes of hands-on time. And then there's an overnight primer binding and extension step. After that overnight protocol, then there's a purification after that, followed by library amplification and a final purification. And at the end, you have your multiplex library ready for sequencing. It's a very simple add and incubate single tube assay. We've automated this assay. We've automated just about all of our assays, by the way, on a number of different platforms. And so if you're scaling up to do more high throughput, we have those scripts and we have those solutions that we can help you with. The kit comes configured with 96 barcodes, although it is scalable and we can provide higher levels of multiplexing as you need them. Our targeted genotyping libraries are sequenced on an Illumina sequencer only, and it uses a custom forward read primer. And then we include the forward read primer, we include all the reagents, all the materials you need to do the targeted genotyping library. So I stole a little bit of my next speaker's slide, but I'm sure he will repeat it and tell it much better than I will. This is a picture of how our library looks. If this is our targeted SNP, we've designed a probe just upstream of that targeted SNP. And our forward read sequence goes directly into the probe. We read the probe sequence, and then we read directly into the region that contains the SNP. And so every on-target read gives you SNP information. The reverse read is where the random fragmented breakpoint is from our random fragmentation, and you can get additional information from the reverse read, either to aid in mapping your reads to the genome or to get additional genotyping information. A couple of things I want to point out. I've already pointed out these two first bullet points. This last bullet point is also important. This design is very flexible. It's very easy for us to produce these primers and these probes. And this is important in case, a year from now, a publication comes out, it's got some great trait that you really want to add, some great markers you want to add into your pool. We can add those into the pool very easily, and you can extend your genotyping pool that way. These are a couple of examples of how the probe location and SNP coverage, how that looks on an IGV viewer. In this panel, this little blue dot here, you see this is the SNP location that we're targeting, and this is the probe. In this first example, there's only a single probe that's targeting this particular SNP. You can see that we've very well covered that targeted SNP. In this case, it's actually a wild type homozygous, and so you don't see any difference in the sequence. But what's interesting is we've identified 2 additional SNPs that can be used as well in this analysis. In this second example, we have 2 different probes that are both interrogating the same target, this targeted SNP here. And so we have sequence information from both directions, and you can see this identified went the wrong direction. I'm going the wrong direction. All right, here we go. identified it as heterozygous SNP. Yeah, the next speaker is actually David. This is one of our customer case studies. And the only thing I wanted to point out on this slide is, as I pointed out earlier, not only will this target enrichment identify the targeted SNPs that you're looking for, but David has shown, and his company has shown, that you can discover new SNPs in the random end of the library in the reverse read. I'll let him tell you more about his design because I don't want to steal all of his thunder. This was a slide I put together because I think it's important to assess. It's important to know how much does it cost, how much does it cost per SNP. And so what I've done is I've put together an estimated cost per data point. These are for a couple of different theoretical designs: a 5,000 SNP design versus a 50,000 SNP design using our approach. Assuming NGS reads of about 300 million reads, 100x sequencing depth, which is way overkill. You probably don't need 100x for doing genotyping. You could probably get away with 25 to 50. But anyway, for argument's sake, that's the number I used. If we multiplex together 540 samples for a 5,000 SNP panel, or 54 samples for a 50,000 SNP panel, sequencing costs are the same, about $2,500 for a sequencing run. Let's say the DNA extraction is $2 per sample. Our library prep costs, what we charge for the kit right now is $25 a sample, up to around $30 per sample for a larger panel design. So the bottom line is looking at the cost per data point. This is a very affordable cost per data point of less than a penny with this panel. Here's a few examples of some of the panels that we've designed up to date: soybean, grape, poplar, tomato, corn, pig, cow, and chicken. And we've got several customers that are looking for new panels as we speak, and we're working on those designs right now. The product does come as a custom configuration, and the cost of the product includes the panel design. So we don't charge anything for the panel design. That's part of the service that we provide with this genotyping product. All right, that's it. Do you have any questions? Happy to entertain any kind of questions for the next few minutes. Yes?
So you're sequencing 100x coverage.
So the 100x coverage, that was simply just for doing a calculation. I think normally for genotyping, it depends on your application, but I think most people would probably do 25x, maybe 50x. I don't think you have to do 100x.
What does it do with the cost if I do 25x?
So at 25x, the cost per data point is even lower. You multiplex higher, so you put more samples in, but the cost per data point is even less than this.
But now it's about $50 per sample. What would it be if I sequence 25x rather than 100x?
So you said it's $50. You said $50 per sample.
Yeah, it's 54 samples for $2,500.
Well, that's the sequencing costs.
Okay.
Right. So the sequencing costs we can't do anything about except for help you multiplex more samples into your sequencing so you get more data out of the run. Our library kit cost is $25. So on top of that, you have to calculate your DNA extraction costs and the sequencing costs, and those are fixed costs that we don't do anything about except for multiplexing. Does that make sense?
Yeah.
Yeah. Yes.
The information for your design panels, let's say for the tomato, is that on your website?
The information about the design for tomato.
Yeah. How many SNPs on the panel, etc.?
I don't remember off the top of my head the design for tomato and how many SNPs that is. These are all custom designs. So we actually, for this product, we don't offer a catalog design at the moment. There may be a point in the future where we would, but just about everybody's kind of got their own panel of SNPs that they want. So the way that works is a customer contacts us, they give us a list of targets that they want to design to. We do a design. We provide the design back just as an electronic file so that they can review it. We'll review it together with them, and once the design's acceptable, we build the kit, and our turnaround time is about 3 weeks to build a custom kit. And as I said, the price per sample on the kit includes all the design. Yeah. Yes?
How much can you increase the multiplexing? Because often breeding company, they need to genotype 1,000 and 1,000 and 1,000 individuals.
Sure.
They even can sacrifice something about quality if they can increase the number of individuals.
Right. Yeah, so we've been talking with customers that want to multiplex up to 3,000 to 4,000, and we have the capacity to make that many barcodes and provide those. We haven't yet had a customer physically literally ask us to do that, but we can do that, and so we will scale that up as the demand grows. That's not a problem.
And surely this reduce the price.
Assuming that would reduce the price, yes. So I think that from what I've talked to customers, $25 a sample is not, that's not a bad price, right? I hope I get some agreement with that. But yes, as you do more highly multiplexing, and as your number of samples increase as well, Nugen, we can work with you on helping you do assay miniaturization if you're doing the right number of samples. It makes no sense to do it for 10,000 samples. Just use this system. Automated system would probably be best. But if you were doing many thousands of samples, I think that's the kind of thing where we can work with you to help you figure out how to reduce costs.